Hi, this presentation on next generation file formats and platforms is a joint production from myself, Josh Moore, from the Open Microscopy environment, primarily talking about file formats, and Nicholas Sofroniew from the Chan Zuckerberg Initiative, primarily talking about platforms, and we've stitched this together. Hope you enjoy. So I'm Josh Moore from the University of Dundee, working for the Open Microscopy environment. Since this video needs to be combined with Nick's, I was actually going to start with my acknowledgements and get that out of the way. And that goes to the current OME team members and past OME team members for making all the infrastructure rebuild possible, but also to the builders of these modern platforms that are using next generation file formats. You've seen Christian Tischer's presentation on Moby, that's using OMEzar. You'll see Nick talk about Napari. There's also Vizar from Harvard Medical School, so thanks to all of them, but also to those who have built the next generation file formats and other file formats for working with very large data. Without them, this wouldn't be possible either. Thanks to those who actually have produced the data. I don't have any data of my own. Everything comes from the IDR. You're welcome to go download it yourselves. And of course, to the funders for making really everything possible. At OME, we focus on building infrastructure to help the community, reuse, access, share their bioimaging data. You can almost think of it at the level of power lines and water pipes. So it's not necessarily sexy and things you can touch immediately, but you realize when they disappear. So what I'd like to focus on for the next few minutes before we get back to the kind of pretty pictures with Nick are what we think is happening at that level of infrastructure, what you need to be aware of, and where the field is going. So the actual products that we build, consist of a data model, a common vocabulary that we can all use to talk about our imaging data. There are a number of file formats like OME-TIFF that you can use to exchange your data. There's the library bioformats, which helps to read hundreds of different proprietary file formats into this common vocabulary. And there's the data management server Omero that you can upload your images to share them and collaborate with others. And we have a fairly good feeling for how much data is flowing into these systems and what they're enabling. At Omero, we know that there are hundreds out there that are running. And from our own use at the IDR, we know basically how much data we can get into an Omero in a given month. So that's order of hundreds of terabytes of data. Bioformats is clearly accessing more data, making more data available to the community. Everything that goes into Omero goes through bioformats. We know that there are tens of thousands of unique users each month. We assume there are orders of magnitude more data flowing through it. And then there's the open file formats that exist in the community at any given time. So in OME-TIFF, that's our charge, is to make as much of that accessible as possible. But then there's all the other data that's out there that's sitting in a format that's not readily accessible. And I guess that's our concern and what I want to bring up here. The organizers, when they invited us, posed this question. I assume it got posed to everyone. You've collected these massive image data, now what, right? I assume at the meeting there have been several different ways of working with this. So you've seen good solutions. What we're concerned about is really kind of the mass sum of this data. So what do we do as a community now that we've collected all this data? Probably one of the biggest concerns is the proprietary data that's out there. So we try with bioformats to make as much of that accessible as we can, but that's getting harder and harder. So we've actually talked to vendors who themselves only test the previous two versions of their own file formats, where we're trying to keep all data that's ever been produced in these formats, that's ever been supported by bioformats, accessible. So how do we solve that tension? How do we get this data open? But even for open file formats, and certainly for the proprietary file formats, they may not be written in a way that that the community can access them freely. So we had this one case of an OME-TIFF, a very large so 500 gigabytes for a single 2D plane. And for whatever reason, and we were quite proud of publishing this in the Journal of Cell Biology, it went viral, and we had thousands of people accessing it, I think every second, but certainly every minute. And the format simply wasn't built for letting that many people access the data. So if our goal is to have all of our data be fair and accessible, even relatively small, on the order of gigabyte files, but certainly as you get into the terabytes and as we go beyond, we really need to rethink how we're making this data accessible to the community. And there have been several groups that have worked quite hard on this. So and developed formats that make it possible to share larger and larger data. So the Big Data Viewer format was developed in Dresden, the Keller lab block file format at Janelia, the Saalfeld lab also at Janelia developed in 5. There's another file format called ZAR. These are all attempts to deal with ever larger data. But then we basically come back to the point that we're going to need infrastructure to then read all of them together. So, if every lab develops their own file format, we're back to square one, and we need to think about as a community, how is it that we balance the evolution of what we're building, the needs of the community, but also having long-term maintainable infrastructure that we can all deal with. And ultimately, with the assumption that you want your data to be accessible, you want it to be fair and shareable in the long term as you publish it. So what I'm going to focus on today are really these bottom two file formats, IN5 and ZAR, which we called next generation file formats. They work fundamentally different from BDV and KLB, or something like HDF5. But the strategies we're talking about can be applied to really all the file formats. And our ultimate goal is to have a small number of open file formats that work very tightly together. So, for the user, it should be completely transparent whether your data is in an OME TIFF or one of these next generation file formats or an HDF5. They should have no loss of data, and they should be able to choose the format that works best for their use case, for their type of data. But since it's less clear what these next generation file formats are, I'll focus on those for the next few slides, give you a sense of how that kind of opens up the possibilities, opens up what Mobi is doing as a next generation platform and what Napari is doing and VZAR. And then I'll give you a bit of perspective about what's possible, and then you'll have to make choices for what you actually want to use. Okay, so next generation file formats, that's another way of saying these are cloud native file formats. They're built for living in the cloud or in object storage. So traditionally, some very large percentage of people have their data locally. They're doing all their research down here in kind of on the file system, on your workstation. And at that point, bioformat still works fairly well. You have your proprietary file format or whatever file format you're working with. You can perform an on-the-fly translation, and you can get it to your analysis routine. As data starts getting larger, you may want to upload that data to a server, for example, for Omero, or that could even be something like Google Drive, not Google Cloud, Google Drive storage or Dropbox. You put your data up somewhere. You're just uploading those large files, and the downloader, the user probably has to download it and do the same on-the-fly translation. Or if it is a server like Omero, there may be on-the-fly translation happening on the server, but somewhere it still has to happen. There's this constant process of rereading the data and unpacking it and trying to get access to it, because those files were built to be written very quickly by the acquisition systems and not really reused over and over and over again by users. So that's really the fundamental change that we're talking about here. What we see as kind of the big one piece of the puzzle that we as a community need to work towards. It's to take, we understand that the file formats generated by the acquisition systems need to be written as quickly as possible, so that will continue to happen. But as a community, when we go to share or especially publish our data, to convert it into an open format that's really intended to be used by users. In the case of the next generation file formats, that means cutting them up into very many pieces, so small little chunks. It's also called a chunk-based format. Uploading all of those to object storage so that each one of those gets their own URL. Making sure that we have multi-resolution levels so that a user has a zoomable experience. So you have the full resolution data, but you also have lower resolution data. And you choose a size for each of those chunks that's really optimal for how your users are going to access their data. So here, saying about 1K by 1K, and the browser can then go in and pull each one of those chunks separately and get, good parallelism. So that's kind of that's the end goal of what we're doing with these new file formats. And to just give you, so I guess a different perspective on why object storage is being sold more and more. So why is it of interest? In a rule of thumb is that object storage on a gigabyte by gigabyte basis will be much cheaper. So it's about 2 orders of magnitude here. So from a euro per gigabyte to a cent per gigabyte, and what you get for that is much more throughput. So that's sounding awfully good. It's cheaper and it's faster. The thing to keep in mind though is that it may be faster, but it's much more. Each access of one of those chunks is much slower. There's more latency, so down from 10 microseconds up from 10 microseconds to a millisecond. So what that means is that the software that's accessing the data needs to be updated to make use of that. It needs to work in parallel. So you can get excessive parallelism with object storage, but you're going to need to plan that into what you're doing. So it's a different way of thinking that we need to be ready for. And the same goes for working at the chunk level. We may be used to writing our data and editing it in place, but each of those chunks is really an atomic unit, and that requires some replanning as well. So those are things to be aware of, and our institutions may be pushing us more and more towards object storage for cost purposes, but keep in mind that there will be a cost at updating our software to work in this way. Hopefully we can all work together to make that happen. So that's certainly that's the infrastructure we're currently trying to build. Okay, so briefly, and I'll hand over to Nick, the take-homes that I see we're talking about moving beyond bioformats. That's bioformats is written in Java. We want access to Python, as in it's happening in Nepari, but we also want to get rid of the overheads. And every time your user accesses the data, that should be as quick and as direct as possible. And to do that, we must go beyond bioformats. In general, think about moving beyond your local boundary. So think about what it means when you start to move your data remotely or into the cloud. That requires a new way of working. It allows more sharing, so there are benefits, but it does require some planning. And beyond bespoke, sorry, I needed another B word. I think this just means it behooves us, another B word, to work together. So each lab shouldn't need to build their own infrastructure. Can we find a common way to build something together? The more that we share our infrastructure, the more utility it has. Think about the power grid, right? So this is how we're working. We're building these various specifications in OME NGFF land. All of that's a public discussion taking place on ImageSC. The specification gets published to ngff.openmicroscopy.org. There's sample data available from the IDR. If you have ideas of specifications you would like to add, or features you need, or other tools you would like to integrate with these next generation file formats, please let us know. We'd love to hear from you. All right. Thank you much. Bye-bye.
Thank you very much, Josh, for giving the first part of our joint presentation. It's really my pleasure to pick up where Josh left off. My name is Nicholas Sofroniew, and I'm the imaging tech lead at the Chan Zuckerberg Initiative. While Josh was telling you about next generation file formats, I'm going to pick up and tell you a little bit about next generation platforms. And so, what do we really mean when I say next generation platforms? I think the best way to describe this is talk about some of the motivations around what's driving some of this development here. And, Josh was mentioning that imaging data sets are getting, larger and larger. And while we actually now do have some really exciting file formats to store that data, it's just as important that our platforms for visualization and analysis keep up pace and allow users to look at and work with and analyze that data too. And so some of the motivations are providing fast, possibly GPU powered visualizations of n-dimensional images and data sets. Data is no longer just 2D, or 3D. It can be volumetric, time series, multi-channels, things like that. Really important to support arbitrarily large data, so bigger than fits in memory. Possibly remote data or even on the cloud. And Josh was talking earlier about some of these cloud access patterns for remote data sets too. And a lot of projects are also thinking about, how to integrate with scientific Python and machine learning ecosystems. There's a lot of really exciting stuff happening in computer vision right now. Automated segmentation methods that are leveraging machine learning libraries in Python, and so a lot of platforms are thinking about that too. And then a lot of these want to be extensible, customizable with sort of easy pathways for installation and discovery of things as well. And then I think it's really important, a lot of motivations to provide inclusive environments and really be community driven too. And so some examples of next generation systems include Neuroglancer, which is a WebGL-based viewer for volumetric data, capable of displaying arbitrary cross-sectional views, 3D meshes, and more from one of the teams at Google. Viv is another a really great library for multi-scale visualization of high-resolution multiplex bioimaging data on the web, and that's from the team at Harvard Medical School. And that integrates really nicely with ZAR, as is Neuroglancer, one of those modern next generation file formats that Josh was talking about earlier. Enjoy is another really exciting platform, plug-in powered hybrid computing. It can work on web and mobile, and it's really around deploying, allows you to deploy deep learning applications or advanced image analysis tools as well. And then Napari is a fast interactive multidimensional image viewer for Python, the plugin ecosystem. And this is a tool that I myself am involved in and work on. And I'm going to talk a little bit more about its capabilities during the rest of the presentation as a sort of illustrative example of some of the things that next generation platforms are looking to provide. And so, Napari, as I said, it's an n-dimensional data viewer in Python. It's open source, community developed. It's got a viewer that will be similar to many imaging applications with fundamental things like canvas, layers, layer controls, dimension sliders, the ability to add, remove layers, control different aspects of the view and status. And we've built this on top of VisPy for our rendering, great Python library, and packages like NumPy, Dask, and ZAR for dealing with really large arrays, and Qt for dealing with the actual interface. And so here we can visualize multi-resolution pyramid data. This is actually an example from Josh and the IDR. It's a ZAR file that we're looking at different resolutions, hundreds of thousands or hundreds of thousands of pixels. We also can do dynamic loading and processing of data from disk. And so in this example, we're taking raw lattice light sheet data and then deskewing that and deconvolving that on demand. And so this is really exciting because maybe you don't want to store 2 copies of this data set, but you're interested in viewing it deskewed and deconvolved. And so each different time point when the slider moves, we load in the necessary data, deskew and deconvolve it as well. Now in Napari we can do more than visualize just images. We have methods for looking at labels, segmentation masks, points, point clouds, vectors, tracks, for tracking objects, services, and more. Now, we also, it was really important to us to enable interactive analysis. And, in this example, you can see someone, using the Python console to access objects that are in the GUI. And you can see someone's, clicking on some points, and then, going back to the console, and now those points are available, the coordinates of those points are available in the console. I think it's, really useful for folks doing a kind of interactive analysis, mixing the GUI and code. As I mentioned, we also really wanted to integrate well with the scientific Python ecosystem. This is an example of a machine learning-based denoising method that's being trained right now, and it's using PyTorch and scikit-learn under the hood as well. Now, we have a plugin model, which is really exciting, and in this example of a cell pose, a deep neural network cell nuclear segmentation plugin from Carsten Stringer at Genelia, and we're able to leverage PyTorch under the hood to do really great segmentation of these cells here. Similarly, Stardust is another really exciting cell segmentation plugin as well. That you can use some pre-trained models here, and then run them and see really great segmentations as well. So with all these plugins, though, we wanted to provide a way to discover, install, and share them. And so the imaging tech team at CZI, in an effort led by Justin Kiggins, built out the Nepari Hub as a place to do exactly that. And you can see here that you can search for and browse individual plugins. Click on them, find plugins with README, so with installation instructions as well. And so, with that, I just want to, bring us back to the idea of next generation platforms and some of the motivations around them. Fast visualizations, arbitrary large data, integration with machine learning, and being easily extensible and customizable are others. And again, really wanting to provide community-driven and inclusive projects. And on Nepari, I want to thank all the folks that have been involved in Nepari development. So really large number of contributors at this point, and we're really excited to welcome and onboard new contributors too. And so if you're interested, do come by the GitHub. We've got a number of places where you can reach us. The ImageSC forum, which Josh talked about before. We have a Zulip chat. We're very active on Twitter as well. We just love to hear from people too. So really, I'd want to thank the organizers again for inviting Josh and myself. It was really fun to do our paired talk, and I hope you enjoyed hearing from both of us today about next generation file formats and platforms. Thank you very much.